Specific immunity is specific to a particular infecting antigen. An antigen being anything that the body recognises as being foreign. And specific immunity has to be acquired. It's not innate. We're not born with it. It's not something that is developed as a result of normal anatomy and physiology. We're exposed to a particular antigen and the immune system mounts a specific immune response to a particular antigen. And there's basically two types of specific acquired immunity. The first is called humoral immunity, to do with the humours and fluids of the body. And the next one is called cellular immunity. But let's forget all about cellular immunity for now and think about humoral immunity. Now, humoral immunity is based on antibodies. The antibodies are the immunoglobulins. And let's draw an antibody to see what's happening. Now, antibodies, there's different classifications of antibodies, but they're all based on a simple Y-shaped molecule. In fact, it's a bit like my body. There's a bit in the middle, and you've got two arms, and the arms can articulate. That's what an antibody is. It's essentially a body. It's a Y-shaped molecule. So we have a Y-shaped molecule here. Protein, immunoglobulin. And this bit tends to be much the same in all antibodies. But the end is very variable. The end of the antibody is a specific shape. So let's draw ours as that kind of shape. That's a particular antibody there. Now, the reason that the antibody is this particular shape is so it can interact with what is called an epitope on an antigen. So here we have an antigen, it could be a virus or a bacteria, and we see that this particular bacteria or whatever it is has epitopes on its surface which are triangular shaped. And what these are actually proteins expressed on the surface of the bacteria. They are highly specific molecules to that particular antigen. So a particular bacteria or a particular virus will have a very specific protein structure. And the part of the antigen that the immune system recognises as being foreign, as not being part of itself, is called an epitope. So these are epitopes on the surface. Now I've drawn them as triangles, but actually they're very complex shapes. And there's many millions of different types of epitopes. So there's tens of thousands of types of bacteria and viruses that can cause infection, all with very specific molecular architecture on the surface of their cell. And they can all cause infections. But we see that this antibody here is the right shape to fit into this particular epitope. Because what the immune system has done is the immune system has made an antibody which is specific to combat this particular antigen. It's a reciprocal shape. So it's really clever. Here's the shape of the epitope molecule. The immune system recognises that and makes an antibody with the equivalent reciprocal shape so it can latch onto it like that. And this means that different bacteria or viruses can be clumped together like this. There's another epitope there, another antibody. And we can see that now we're starting to clump together lots of these nasty bacterial cells. So the antibodies are acting against these bacteria or viruses, clumping them all together. And this is going to inactivate them, and also it puts them all together in one place. Instead of running wild all over the place, they're all nice together. They're agglutinated into a particular clump. And that means a nice big phagocytic cell like a macrophage can come in and eat them all together. So it's sometimes said that the antibodies prepare the food for the phagocytes table. They get it all together so it's ready for them. So the key to humoral immunity is that the immune system produces millions and billions of these very specific molecules called antibodies. They're released into the blood and the tissue fluid. They circulate around and they combat the antigens. And this is one way that it combats them. But the thing is, of course, where do these antibodies come from? Well, the antibodies are actually produced by the B lymphocytes. The B lymphocytes actually produce these antibody molecules. Now, what you have on a B lymphocyte, here's a B lymphocyte, and the actual antibodies to begin with are on the surface of the B cell, so they're kind of sticking out from the surface of the B cell like this. There's the antibodies. Now, we've said that there's millions of different types of possible molecules on the surface of antigens. Well, in the same way, there's actually millions of different types of B cells. They're all B lymphocytes, but they'll have a different shaped antibody on their surface. So it will recognise a different type of antigen. So, for example, you might get a B cell here, and this B cell has an antibody on its surface, and it's a circle shape, like this. So that would recognise circle-shaped antigens should they come along on a particular microbe, particular antigenic bacteria or virus. So really what we've got is many millions of types of B cells. They're all B cells, they're all B lymphocytes, but many different, expressing many different molecules on their surface, and many different potential infecting antigens. And what actually happens when a particular antigen comes into the body, it might try thousands of B cells before it gets the right one, but eventually it'll get the right one. And what we say is the B cell recognises the particular antigen. It's kind of a selection process. We can sometimes call this B cell selection. So out of the millions of different B cells in the body, maybe only one or two cells with the right shaped antibody on its surface bind onto the particular antigen that's causing the infection at the time. Now, when that happens, when the specific antigen that fits onto the antibody on the surface of the B cell, when that fits together, what this does is it activates the B cell. So once the B cell has been selected by this antigen, the B cell is activated. And what the B cell then does is it divides into two. Now you've got two B cells instead of one. But this B cell, these daughter B cells, have got exactly the same arrangement of DNA as the parent B cell, so they make exactly the same shaped antibody. And then they'll divide again. So now we've got 4. And they'll divide again until we've got untold millions of them, enough to increase the total white cell count in the blood. This process is called clonal expansion. What is a clone? Do you remember what a clone is? Well, a clone is any two organisms with the same genetic material. So the daughter cells have got the same genetic material as the parent cell. They're clones. So you end up with a great big population of clonal B cells, all expressing the same type of antibody on their surface. But as this expansion takes place, what happens is that the B cells actually differentiate somewhat into cells called effector cells. So here we have an activated B cell. And this activated B cell line here changes into untold millions of activated B cell clones. And these actually change into cells called plasma cells. They've got very extensive endoplasmic reticulum and produce lots and lots of antibodies. And they secrete these antibodies from the surface. Now, unlike the parent B cell, where the antibody was fixed onto the surface, these antibodies are simply released from the plasma cells into the body tissue fluids. So the B cell is producing millions, billions of antibody molecules. So let's just review that. Antigen comes into the body with a particular epitope on its surface. That's recognised by a very small population, maybe only 1, 2 or 3 B cells, with an antibody on its surface which happens to be reciprocal to that antigen. This particular antibody is able to combat this particular antigen, but we've only got a couple of cells in the whole body maybe with that particular shaped antibody. So this cell is activated and divides into clones of millions of them. So we get the B cell here, activated by this process of clonal expansion, differentiating into effector cells called plasma cells, releasing untold billions of antibodies, and these antibodies then combat, they fight the original infection, the original antigen. And as well as that, the B cell differentiates into another population of cells, and these are called memory cells. They're also clones. You won't get as many of these made, maybe a few thousand of these are made. So you might get many millions or billions of these made, and a few thousand of these made. These get rid of the infection by producing the antibodies. But the memory cells and their descendants stay in the circulation for years or decades. And this means if the same antigen comes along to cause a secondary infection, you've already got a very large population of memory cells which can undergo rapid clonal expansion into another population of plasma cells to get rid of the infection much more quickly next time. But immunity is always a bit more complicated than that, although this is true. What actually happens is that you need the T cell, remember the T lymphocytes, and particularly a T helper lymphocyte, to stimulate the B cells by release of a cytokine, in this case called interleukin type 2. So the T cells also recognise the presence of antigens. They will also proliferate. The T helper cells will then release cytokines stimulating the B cells. The B cells will undergo clonal expansion, produce millions of plasma cells. They'll produce the antibodies and they'll combat the infection. But then, when the infection's gone, it's time to stop this proliferation of B cells. So when the infection's gone and it's time to limit this particular immune response, another type of T cell, this time called a T suppressor cell, will also send cytokine signals to the B cell saying, that's okay, you've done your job now, stop clonal expansion, stop proliferating, stop producing all these antibodies, and that's the end of that particular infection. Apart from the fact we have the memory cells, several thousand of them, which can respond to that antigen much more quickly should there be a secondary infection. So antibodies are immunoglobulins, immune proteins. And these are soluble proteins that go into the blood and the tissue fluids. They're a type of gamma globulin. Immunoglobulins are gamma globulins and they're abbreviated to Ig, immunoglobulin. Now, there's protein, as you know, in the plasma. Most of this is albumin, but about 20% of plasma proteins are these globulin, immunoglobulin type proteins. And you might have come across a blood test where we measure these in the blood, the amount of gamma globulins in the blood. And a specific immunoglobulin or a specific antibody, after the activation of B cells, will be produced. And then they're detectable in the serum a few days after that production by the B cells. So the B cells are activated, as we've seen. The B cells produce the antibodies. The B cells are activated by an antigen. That causes the production of the antibodies, and they can be detected in the serum a few days later. And in fact, the affinity of the antibody to a particular antigen improves as the response goes on. So the B cell actually learns to make even more specific antibodies that are even more specific to the shape of the particular antigen. Because it's actually more complicated than we looked at before. I mean, that's an antigen cell there. The epitope actually might be quite a complicated shape, something like that. Much more complicated molecular shape. So the antibody needs to be made, which is a reciprocal shape to that. That would be the arm of the antibody. And it's made more specific. The actual shape, the molecular shape of the antibody itself is made more specific to the antigen as the immune response develops. It's very important that these can migrate across the placenta, because this allows newborn babies to be immune to everything that mum's immune to, and into the colostrum and into the milk as well. And as we've seen, antibodies will agglutinate pathogens. They will clump them all together. They'll precipitate them all together. And if there's soluble antigens, such as tetanus toxin in the blood or the tissue fluids, again, what the antibodies will do is lots of antibodies. They're going to actually stick together the soluble toxins in the blood. So there's our soluble toxins causing havoc. And groups of antibodies will stick them all together. They'll be agglutinated and therefore precipitate out of solution, because they'll now be big molecules. And whenever we get a load of toxins, or indeed a load of pathogens, agglutinated together by antibodies, we call that an immune complex. So that's an immune complex there. Or if there's lots of bacteria or viruses, again, all clumped together by the antibodies. Let's draw the antibodies red this time. Clumping them all together. That also is an immune complex. An immune complex is a load of antigens clumped together by antibodies. And antibodies can also neutralise toxins covering toxic sites. So we might have an antigen here, and there might be a toxic site on its surface like this that can damage body tissues. Well, the antibodies can cover that up, preventing it from being toxic. And antibodies can also act as opsonins. Now, opsonins are immune labels. They will label antigens as being foreign so that the phagocytes can recognise them and then phagocytose them. We've already looked at that process previously. And there's also antibody-dependent cytotoxicity. Now, cytotoxicity is carried on by killer cells such as T cytotoxic cells. And what actually happens here is we'll have a T cytotoxic cell there, like that. A T cytotoxic cell is a cell which can kill other cells, and it has a particular receptor on its surface like this. Not drawn to scale, obviously. That's actually called an FC receptor. And what happens is that the antibody will fit into that. There's the antibody molecule fitting into that. And then this will actually fit onto the epitopes in a particular bacterial cell, or whatever it happens to be causing the infection. And the antibody, of course, and the FC receptor are very small. So what this means basically is that the T cytotoxic cell is now in pretty well direct contact with the infecting cell, and it will release chemicals onto it to kill it. such as perforin, which makes holes in the cell membrane, as we saw demonstrated with the balloon. And the immune complexes that we've talked about, the combinations of antigen and antibody, activate complement. And again, we've talked about complement already, but complement, the end product of the complement process, you might remember, is membrane attack protein, which attacks, perforates and kills bacterial cells. And complement has various other immune functions as well. So a few more functions of antibodies there if you wanted a bit more detail.